Greetings to Dr. Eng and hi everyone. In this video, we will be explaining about enzymic catalysis. So enzyme is actually used to describe a biological catalyst where enzymes are named by adding the suffix "ase" to the name of the substrate that they modify or the type of reaction that they catalyze. Enzymes are proteins and usually have a globular tertiary structure where the shape is determined by the sequence of amino acids. Thus, they are highly specific to the reaction that they catalyze. Enzyme increases the rate of reaction by lowering the activation energy. Enzyme also can work in either direction as long as the substrate and the product form are in equilibrium. Enzyme also used in a very small quantity because they can be reused since enzymes are not destroyed or altered throughout the reaction. However, enzymes are very sensitive to pH changes and also excessive heat exposure because such condition will cause the enzyme to be denatured. Enzymes are much more specific in the reactions they catalyze than inorganic catalysts. Most intracellular enzymes work on a particular substrate while some digestive enzymes work on a comparatively wide range of substrates that are related. So how does the enzyme work? Substrate will bind on the active site of the enzyme, which will cause the formation of enzyme-substrate complex. And once the products are formed, the products will be dissociated or separated from the active site of the enzyme. So as per the lock and key model, the model actually explains the specificity of enzymes where both the enzyme and the substrate possess specific complementary geometric shapes that fit exactly into one another. But for the induced fit model, even though the substrate has different shape that doesn't match with the shape of the active site, it's not a big deal because the active site will continue to change until the substrate is completely bound.
Hi, my name is Norizzati Maisarah, Binti Hanafi, and I would like to talk about the functions of enzyme catalysis and their applications. The functions of the enzyme catalysis are oxidation and reduction, hydrolysis, group transfer reaction, removal of a double bond with a group transfer, isomerization of functional groups, and single bond formation by dehydration. For the function of oxidation and reduction, the oxidoreductases are the enzymes that carry out redox reaction. You can see the ethanol converted to acetaldehyde and cofactor while nicotinamide adenine nucleotide NAD is converted to NADH. For the hydrolysis, you can see that hydrolysis are the enzymes that break single bond by adding the element of water. For example, phosphates break the oxygen-phosphorus bond of phosphate ester. For the function of group catalysis, which is a group transfer reaction, the transferases are enzymes that move functional groups from one molecule to another. We can see the alanine aminotransferase shuffles the alpha amino group between the alanine and aspartate. For the function of removal of double bond with group transfer, the functional group transfer by this layers enzyme include amino group, water, and ammonia. The first application that dehydratase remove water as in fumarate. And for the second application, you can see the deaminase remove ammonia in the removal amino groups from the amino acids. Next, the function of isomerization of functional group. You can see the enzyme, which is the isomerases, as the triose phosphate isomerase carry out the rearrangements in the glyceraldehyde triphosphate to the hydroxyacetone phosphate. Lastly, the single bond formation by dehydration. This function we can see in the application when ligases carry out the converse reaction, removing the elements of water from two functional groups to form a single bond. The synthetases are a subclass of ligases that use the hydrolysis of ATP to drive this formation. Amino acid transfer RNA synthetase join amino acid to their respective transfer RNA in the preparation of protein synthesis. You can see in the action of glycyl tRNA synthetase. That's all from me. Thank you.
The middle of the 20th century saw an exponential increase in the research on enzymes. Therefore, the classification and the cataloging of enzymes became a necessity. Enzymes can be grouped according to the following considerations. Firstly, the enzymes can be grouped according to their occurrence and the source of enzyme. For instance, papain. It is an proteolytic enzyme that is extracted from the raw fruits of papaya to break proteins into amino acids and peptides. On the other hand, horseradish peroxidase is an enzyme extracted from the roots of horseradish. So it catalyzes the oxidation of organic substrates by hydrogen peroxide. On the other hand, the enzymes can also be classified according to the nature of substrate. For example, the enzyme urease catalyzes the conversion of urea into carbon dioxide. The enzymes can also be categorized based on their cofactor requirement. For instance, for thiamine pyrophosphate enzymes, we can say that pyruvate decarboxylase, pyruvate dehydrogenase, and transcriptolase are TPP enzymes because they utilize TPP as a cofactor in the catalysis reactions. Well, this diagram shows the fermentation of alcohol by pyruvate decarboxylase, and this is the enzyme that requires TPP as the cofactor. Next, we can also classify the enzymes based on common functional context. For examples, we can group the enzymes as the enzymes of histidine biosynthesis, or we can group certain enzymes as enzymes that belongs to mitochondria. On the other hand, enzymes can also be grouped according to the nature of overall reaction. For instance, enzymes that catalyze hydrolysis or enzymes that catalyze oxidation. This type of classification is the most commonly used classification. It is a systematic classification used by the Enzyme Commission. As shown in the diagram, the enzymes are classified according to 6 groups of functions. The first one is hydrolases, transferases, oxidoreductases, ligases, isomerases, and lyases. Each enzyme has an EC number that consists of 4 numbers, and they are unique to each of the enzyme. So next, the enzymes can also be classified according to the mechanism of the reaction. We can consider the mechanism at the active site or the intermediate complexes. For example, proteases can be classified depending on whether the enzyme-bound acyl enzyme intermediate is formed or not. So that's all for the classification of the enzymes. Thank you.
Then I will talk about the 5 factors influencing enzyme activity. In the activator effects, there are 2 terms, activators and inhibitors. From the name, we know that activators will let the reaction become faster, while the presence of inhibitors will inhibit the reaction that usually used in the medical field to inhibit the microorganism activities in human body. In the pH effect, different enzymes have different optimum pH range to stay active and stable. Because pH can affect the ionization state of acidic or basic site of enzyme. There are some examples in the table that we can be referred. Next will be the temperature effect. Same with the pH effect. Different enzymes have different optimum temperature. Above the optimum temperature, it will increase the strength to the bond and resulting in break bond in enzyme. So the shape of the enzyme will be changed. Next will be the pressure effect. Increase in the pressure will decrease the enzyme activities until the complete inactivation because the same region of the bond will be disrupted. Hence, by controlling the pressure and the temperature of enzyme can be inactivated. Lastly will be the concentration effect. Increase the concentrations will increase the enzyme activities. However, when it is concentrated, the maximum of the enzyme reactions rate is the highest rate of the whole reactions. There are some references that we use in this topic. Thank you.